Hello, everybody. Welcome back. We don't have anybody in yet, but hopefully we'll get a couple of people. Today we are doing chapters 14, 15, and 16 and reviewing for the test for anybody who shows up. Who we're waiting for. Hello, Kiara. How are you?
Hi.
Glad you could make it. In addition to chapters 14, 15, and 16, we're doing review for the this week's exam. So if you have any questions about the review questions, that's what I'm here for. So we'll do that at the end.
I'm gonna put you on mute. My son's out here.
Okay. I'm going to go ahead and get started. We're starting with chapter 14. And so go ahead and get your lecture notes out. And here we go. Here's chapter 14. Okay, we're talking about nervous tissue here. Okay, the nervous system. The nervous system is the body's primary communication and control system. It's the quick communication system. The endocrine system is the slower system. The nervous system can be divided according to both structural and functional categories. So this is key for the exam when you're looking at review questions associated with this material. You want to make sure that you understand that there are several different ways you can divide the nervous system. And one of the answers to one of these questions may be all of these are correct. So for everybody who's listening, please remember that when offered the choice, all of the above, all of these answers are correct, is usually the right answer when offered. But none of these answers are correct is almost never the right answer. Because I don't put all of these answers are correct as an option unless that's right. Maybe 1 out of 100 times it's the wrong answer as a distractor. Okay, so keep an eye on that. When you see that option, there should be a very good reason for you to reject it, not just because it doesn't look familiar. I'm asking you to use a little bit of critical thinking with these exams, so please be a little bit better with that if you can. Okay. Structural organization, structural subdivisions of the nervous system include the central nervous system, or CNS, which includes the brain and spinal cord. The peripheral nervous system is all nervous tissue outside the brain and spinal cord. That includes cranial nerves, spinal nerves, and ganglia found normally in the back of the body. Okay, this shows you the nervous system. Interestingly, the eye is part of the nervous system because the retina contains nerve cells. It's considered part of the nervous system, even though it's also part of your special senses. Okay, functional organization. So instead of just structural, what does it do? The functional divisions of the nervous system include the sensory, afferent. Please note, sensory or anything coming in towards the brain is considered afferent, and efferent is anything going out in terms of motor. Receives sensory information or input from receptors and transmits it in the form of information to the CNS. Normally, this information is found coded in the form of action potentials. So the motor or efferent division is the outgoing signals, basically telling your body what to do. So from muscles or glands to muscles or glands from the CNS would be the motor or efferent division. So you can see the nervous system is divided into both sensory and motor, and then the sensory is divided into somatic and visceral. So somatic would be what you can feel consciously, visceral would be subconscious, and then the motor division, same thing, your skeletal muscles, and then all the other muscles and glands associated with that. Okay, for the sensory division is subdivided into 2 components. So note that there's many different levels of divisions here. So you got to keep those straight in these review questions. Somatic sensory components are the general somatic senses: touch, pain, pressure, vibration, temperature, and proprioception. Proprioception has to do with your ability to sense where your body is in space. Visceral sensory components transmit nerve impulses from blood vessels and viscera to the CNS. The visual senses primarily include temperature and stretch. These would be involuntary or non, there's a word I'm looking for here and I lost it, unconscious, the word I'm looking for. Okay, the motor division, this would be the division that sends signals outward towards the body. It's divided into two components. The somatic motor, somatic nervous system, conducts nerve impulses from the CNS to skeletal muscles. It's the only motor division that is conscious. It's known as the voluntary nervous system. So there's conscious and subconscious, voluntary and involuntary. So this would be the voluntary. The autonomic motor component, autonomic nervous system, or ANS, innervates internal organs, regulates smooth muscle, cardiac muscle, and glands. It's known as the visceral motor system, or involuntary or subconscious nervous system. Nerve cells themselves, there's two distinct cell types that form nervous tissue. The first that we're most familiar with are neurons, what we call nerve cells. They're excitable cells that initiate and transmit nerve impulses. These nerve impulses are usually in the form of an action potential. But glial cells, or neuroglia, are also a really important part of the nervous system, and they're non-excitable. That means they don't send action potentials, but their key thing is to support and protect the neurons. The analogy I like to use with this is that the neurons are kind of like queen bees in a hive, and the glial cells are more like worker bees. There's a lot fewer neurons, a lot more glial cells. They do the tough work to keep the neurons alive and to guide their repair if they're damaged. What are some characteristics of neurons? They have a high metabolic rate. That means that they use a lot of energy. So when you've been studying hard and you feel tired, it's true. You have been using a lot of ATP, a lot of energy to think hard. Neurons have extreme longevity. They live a lot longer than most cells. And why is this so significant? Because nerve cells, or neurons, are really stretched out, and it's very difficult for them to divide through mitosis. So they tend to have a longer lifespan than cells. They're typically non-mitotic, meaning they can't divide, mostly because their really stretched out nature doesn't lend itself very well to mitosis. What's some of the structural components of neurons? Neurons come in all shapes and sizes, but neurons share certain basic structural features. A typical neuron has a cell body in the center, dendrites coming in towards the cell body, and axons. The cell body serves as the neuron's control center and is responsible for receiving, integrating, and sending nerve impulses. So in this way, the neuron cell body is kind of like the brain of the neuron. It does the thinking and decision-making for that individual cell. Neurotransmitters like acetylcholine are synthesized in the cell body and then sent out usually through axonal transport down towards the axon terminus. Dendrites. Dendrites tend to be shorter, smaller processes that branch off the cell body. Many neurons have only one dendrite, while others have many. The key to dendrites is they bring incoming signals towards the cell body. Dendrites conduct nerve impulses toward the cell body. They receive input and then transfer it to the cell body for processing. Usually that's in the form of a graded potential, which can initiate an action potential, details of which I'll explain to you if you take my physiology course. More dendrites a neuron has, the more of nerve impulses that a neuron can receive from other cells. That is in the form of dendritic transmission, but some impulses can come in the form of axonal transport as well. Okay, the axon, the larger, typically longer nerve cell process emanates from the cell body. It's called the axon, sometimes called the nerve fibers. That's another name for it. So remember, on my test, sometimes there's two answers for something, axon and nerve fiber. Most neurons have only one axon. The axon transmits nerve impulses away from the cell body, always towards another cell. Good way to remember this is axon starts with A, away starts with A, they match. Okay, this is an image of a typical peripheral neuron. You can see the cell body here in the center. The dendrites are surrounding it, bringing cell signals in towards the cell body. There's the nucleus. And then the axon, often wrapped in myelin. It's like a light blue material here, insulated. And then down here you have the axon terminus with the synaptic knobs sending signals out themselves. This is what a ox spinal cord neuron looks like. You can see the cell body, the axon hillock, where the axon attaches to the cell body, and then the axon moves its way over from here. So this is what it would look like under the microscope. What are some more classifications of neurons? Well, they vary widely in morphology and location. Morphology means shape. They can be classified according to either their structure or their function, as we've seen. Neurons can be classified according to the number of processes extending from the cell body. So pole means a place above or below the top of the cell. So unipolar has a single process. Bipolar, you've probably heard that term before, but in this case, it means having one process on top and one on the bottom. And then multipolar have many processes. So here is a unipolar. You can see only a single process here sticking out. There's bipolar, having one on either side. And then multipolar neurons are down here, lots of processes emanating from the cell body. Interneurons. Okay, interneurons, or association neurons, lie entirely within the central nervous system and are always multipolar. Now, in the same way we talked about that certain parts of the nervous system can be either sensory or motor, well, neurons can also be sensory or motor. Interneurons, though, are neither. They don't bring signals in and they don't send signals out. They are really the thinking cells. They connect cells to each other and make decisions within the central nervous system. They receive nerve impulses from many other neurons and carry out the integrative function of the nervous system, that is, decision, memory, stuff like that. Thus, interneurons facilitate communication between sensory and motor neurons. So the sensory brings signals in, interneurons make the decision, and then send the signal to the motor neurons, which then tell the muscles and glands what to do. So you can see from the skin, afferent transmission. In this case, afferent doesn't mean away, it means towards the CNS. You can see the posterior root ganglion. The reason why it's swollen is because the cell bodies are located here, making it thicker. And you can see an interneuron here in the spinal cord, which is part of your central nervous system. This makes a decision about what's going to happen as a result of the signals coming in from the skin. If the signal is strong enough, it sends out impulses to the motor neuron, here shown in red, and then out to the muscles. This shows you some very different kinds of cells that you would find in the brain: astrocytes, epidermal cells, oligodendrocytes, etc. Okay, let's continue on with chapter 14. Glial cells, sometimes they're called neuroglia, occur both with the central nervous system and the peripheral nervous system. Glial cells are smaller and are capable of mitosis. That means they can divide. Glial cells do not transmit nerve impulses in the form of action potentials. Their sole job is to support and nourish the neurons. Glial cells physically protect, help nourish neurons, provide an organized supporting framework for all nervous tissue. Glial cells far outnumber neurons, so they're smaller, but there's a lot more of them. Glial cells account for roughly half the volume of the nervous system. How would you account for that? Well, if they're much smaller, you would need many more of them to get half of the volume of your entire nervous system. And this shows you an astrocyte, which is a glial cell. These are epidermal cells, another kind of glial cell. Here's a microglia, a kind of a white blood cell, so to speak. And oligodendrocytes wrap the axons of the central nervous system, especially the brain, insulating those axons from leakage, make sure the signal spreads from one place to the other without loss. Okay, this is the posterior root ganglion, the satellite cells. The satellite cells is what we're talking about here, as well as the wrapping cells, even though they're not labeled. And here we're talking about the Schwann cells, which are these light blue cells that wrap the axon in the peripheral nervous system and insulate the signal, as we talked about earlier. Glial cells of the central nervous system, astrocytes, exhibit a star-like shape. So think of astro and star. Due to projection from the surface, astrocytes are the most abundant glial cells in the central nervous system and constitute over 90% of the tissue in some areas of the brain. So that's a lot. They help form the blood-brain barrier, which is abbreviated BBB, that strictly controls substances entering the nervous tissue in the brain from the bloodstream. So this is like a gatekeeper to make sure nothing inappropriate goes into your brain. And it can help regulate tissue fluid composition. What are some more glial cell functions? They can form a structural network. They can replace damaged neurons. They can assist in neuronal development, that is in fetal development. They can help guide the movement of axons as they're moving to make connections with other cells. What is myelination? Well, myelination is the term that means Schwann cells or neuroleukocytes can be wrapping around a neuron or axon so that it can make sure the signal remains within the axon, doesn't leak out into surrounding tissues. Myelination is the process by which part of an axon is wrapped with a myelin sheath, a protective fatty coating that gives it a glossy white appearance. That's why part of your brain and spinal cord is called white matter, and some is called gray matter. The white matter is myelinated axons. The myelin sheath supports, protects, and insulates an axon, the most important function being the insulation, making sure the electrical signal does not leak. No change in voltage can occur across the membrane in an insulated portion of an axon. What it does is it makes sure that no voltage leak occurs in that part of the axon. In the peripheral nervous system, myelin sheaths form from neuroleukocytes. In the CNS, they form from oligodendrocytes. Now, how do you say a big word like this? Break it up into little pieces. Oligodendrocytes. That's how you get those big words pronounced. Okay, so this shows you how myelination occurs. We have the neuroleukocyte beginning to wrap around the axon, and it wraps around over and over. The key is it's increasing the cell membrane, which is fatty, phospholipid, and decreasing the amount of cytoplasm. As you can see, as it wraps over and over and over again, removing cytoplasm but increasing cell membrane, you end up at the very end with this myelin sheath, which is all of this fatty material, which is very electrically insulated. Okay, here is showing you the central nervous system, that oligodendrocytes, and each of these cells here are neuroleukocytes or Schwann cells in the peripheral nervous system. Myelinated versus unmyelinated axons. In a myelinated axon, the nerve impulse jumps from neurofibril node to neurofibril node, and this node is saltatory conduction. In Spanish, saltar means to jump, so if you know Spanish, it might make it easier. In an unmyelinated axon, the nerve impulse must travel the entire length of the axon, a process called continuous conduction. Now, the analogy here is, what is faster, a kangaroo walking or a kangaroo jumping? Obviously, a kangaroo can jump far faster than can walk. And so, in a myelinated axon, the signal jumps from node to node, being much faster than walking along an unmyelinated axon. So myelinated axons produce a faster nerve impulse. The exact reason why this happens, I'll tell you about in physiology. In an unmyelinated axon, the nerve impulse takes longer to reach the end of the axon. A myelinated axon also requires less energy in the form of ATP than does an unmyelinated axon. Using continuous conduction, unmyelinated axons conduct nerve impulses from pain stimuli. So pain receptors tend to be more primitive, unmyelinated, slower conduction speeds. So this shows you how in some parts of the nervous system, you can have several axons being surrounded by a single neuroleukocyte. This image shows you how when an axon is damaged due to an injury, the distal portion disintegrates, and then the neuroleukocytes help guide the axon moving its way down back towards the muscle that it would be stimulating. And as you can see, this is the end of it, and eventually it makes its way back. The regrowth speed in most cases is about 1 inch a year, which seems really slow, but it's such a small cell and it has to go through such detailed, extensive growth. That is pretty quick for that kind of cell. So regeneration of a peripheral nervous system axon. PNS axons are vulnerable to cuts, crushing injuries, and other trauma. Why so much more so in the peripheral nervous system? Well, think about your arms and your legs. tend to be out there and more of a danger. The central nervous system is within your skull, within your spinal, your vertebral column, tends to be more protected. So peripheral nervous system axons are the ones that are most likely to be damaged. A damaged axon can regenerate, however, if at least some of the neurolemma remains. And what does that mean? The neurolemma acts kind of like a stem cell, helping reformation of the axon back towards its terminus. The peripheral nervous system axon regeneration depends on three main factors: the amount of damage, neurolemmacyte secretion of nerve growth factors to stimulate outgrowth of severed axons, and the distance between the site of the damaged axon and the affected organ. As I said, 1 inch per year. Greater distance takes more time. Structure of a nerve. Now, please note, nerve and a neuron are not the same thing. A nerve is a bundle of parallel axons. So an anatomical nerve that you find in your arm is not a whole bunch of neurons, it's just axons bundled together like a cable. Like a muscle, a nerve has 3 successive connective tissue wrappings. The endoneurium is a delicate layer of loose connective tissue that wraps an individual axon. The perineurium is a cellular and fibrous connective tissue layer that wraps groups of axons into bundles called fascicles. And an epineurium is the outermost layer, a superficial connective tissue covering made of dense, irregular fibrous connective tissue, which encloses the entire nerve, providing both support and protection. This shows you what a cross-section of an anatomical nerve would look like. As you can see within the nerve there are these bundles. Each of these bundles is called a fascicle. Within the fascicle are individual axons surrounded by the myelin sheath and the endoneurium around each axon, and blood vessels providing food and oxygen. Some more images of these same structures. Nerves are a component of the peripheral nervous system. There are no nerves per se in your brain. It's just primarily associative neurons. Sensory or afferent nerves convey sensory information towards the CNS. And motor efferent nerves, or neurons, convey motor impulses from the central nervous system to the muscles and the glands. Axons terminate as they contact other neurons, muscle cells, or gland cells. An axon transmits a nerve impulse at a specialized junction with another neuron or another cell. It's called a synapse. Synapse means a spot where an axon touches another cell. So what do you know about synapses? Well, the presynaptic neurons transmit nerve impulses along their axonal membranes toward a synapse. So it goes from the cell body down the axon towards the axon terminus, and at the axon terminus is a gap called a synapse. The postsynaptic neuron, or postsynaptic cell, conducts nerve impulses through their dendritic and cell body membranes away from the synapse. Now, this would only be the case if the postsynaptic cell is a neuron. If it were a muscle, it would be different. And we'll talk about the differences between action potentials in muscle cells and action potentials in neurons in physiology. Axons may establish synaptic contacts with any portion of the surface of another neuron except those regions that are myelinated, because the myelin is insulation, no way to contact a cell to transmit information, electrical information. So this shows you at the bottom a highly stylized version of a presynaptic and postsynaptic neuron, with the synapse being a gap. And this is the more accurate diagram of synaptic points from the end of one axon to the cell body or dendrites of the second cell or postsynaptic cell. And you can see here a little bit more detail showing you the insulated axons and how potentially they could interact in places called telodendria. I'm not going to go into a lot of detail with this. Here it's called axosomatic synapse, somatic meaning body, in this case cell body. Axodendritic synapse, axon to dendrite, and then axoaxonic is axon to axon. Electrical synapses. Electrical synapses are not very common in mammals. Turns out the most common is chemical synapses. In humans, these synapses occur primarily between smooth muscle cells, where quick uniform innervation is essential. Electrical synapses are also located in cardiac muscle. The most numerous type of synapse is the chemical synapse. It facilitates most of the interactions between neurons and all communications between neurons and effectors like muscle cells, glands. At these junctions, the presynaptic membrane releases a signaling molecule called a neurotransmitter, such as acetylcholine, abbreviated ACH. Other types of neurons use other neurotransmitters, which I discuss in physiology. So this is an image showing kind of how it works, the details of which I'll go into in physiology. Fundamentally, when a nerve impulse comes down in the form of an action potential, changes the surface electrical potential that causes calcium gates to open. Calcium causes these bubbles, called vesicles, to dock here at the synapse, dumping their acetylcholine. Your acetylcholine sticks to receptors on the postsynaptic cell, opening sodium gates and therefore allowing a greater potential to build up and eventually another action potential if the signal is strong enough coming from this neuron. What are some different neurotransmitters? Well, they're released only from the plasma membrane of the presynaptic cell, not the postsynaptic cell. It then binds, when we say it, the neurotransmitter molecules bind to the receptor proteins found only on the plasma membrane of the postsynaptic cell. This ensures a one-way flow of information from presynaptic cell across the synapse to the postsynaptic cell. A unidirectional flow of information and communication, therefore, takes place. Two factors influence the rate of conduction of the impulse: the axon's diameter and the presence or absence of a myelin sheath. We mentioned earlier, presence of a myelin sheath increases synaptic transmission speed. Okay, that is the end of 14. So now we are going to go to 15. Kira, do you have any questions before we continue on? I'll take that as a no. Okay. Here we're talking about the brain and cranial nerves. An adult brain weighs between 1.35 and 1.4 kilograms, which is about 3 pounds. It has a volume of about 1,200 cubic centimeters, called cc's. Now, interestingly, the size of a person's brain really doesn't determine their intelligence. It has more to do with the connection between brain cells. But if you're comparing different species, you can say something about intelligence. So, for example, because a human's brain is bigger than a dog's brain, human beings have a better ability to process information and make decisions. Doesn't mean those decisions are going to be good ones. I think dogs make better decisions than people do. But just because a person has a bigger head than somebody else, and therefore a bigger brain, doesn't make them any smarter. Brain size is not directly correlated with intelligence. It has to do with connections, and the connections has to do with learning and keeping your brain active. It's not the physical size of the brain that determines intelligence. It's the number of active synapses. So use it or lose it. What are the four major regions? Well, the cerebrum, the diencephalon, the brainstem, and the cerebellum are the four major regions of the brain. The cerebrum is divided into two halves called the left and right cerebral hemispheres. Each hemisphere is subdivided into 5 functional areas called lobes. The outer surface of an adult brain exhibits folds called gyri, which is plural, gyrus is singular, and shallow depressions between those folds called sulci, plural, or sulcus, singular. The brain is associated with 12 pairs of cranial nerves. Now, what I may ask you about on an essay is, what does this image have to do with an image I'm going to show you later? Well, if you look at the surface of a human brain and you stimulate it, the person will believe that you are actually stimulating these body parts. So along this area of the brain, if you get stimulated, You feel it in your hand, and that means this part of your brain is dedicated for processing signals coming from your hand. Notice so much less coming from the rest of your body. There's a whole section for your face, the lips. Look how big the lips are, and then tongue, etc. Here. So this is the sensory homunculus. This is the motor homunculus, dedicated towards sensation and dedicated towards movement. Now notice, not a lot of movement in the genitals, but there is sensation in the genitals, much smaller than you would expect. Well, sensitive parts of the body, believe it or not, are the feet, the hands, and the lips. Genitals, well, when they're touched, it's very significant. They're not as sensitive as you might think, especially when compared to the hands and lips and feet. So if you put all those images together, you get what is called the homunculus. So what this homunculus is showing is how much of the surface of the brain is dedicated to these sensory parts of the body. Notice how big the lips and tongue are, how big the hands are, and how small the rest of the body is. Look at even the ears are pretty large. Eyes are fairly large. But look at how small the rest of the body is. That means you have far fewer sensory cells and therefore less of the brain dedicated to that part of the body than the hands and the lips. Interestingly, we use our hands to touch things and identify them. Therefore, we want them to be very sensitive. Okay, moving on to the different lobes. The frontal lobe is responsible for consciousness and judgments, how we initiate activity in response to our environment. It controls our emotional response, controls our expressive language, assigns meaning to the words we choose, abstract thought. It's responsible for attention span. So when they say a young child's frontal lobe is not fully developed, that's what they mean. They can't pay attention as well as an adult, hopefully. Involves word association or language planning. What words are you going to be using in the future? Memory for habits and motor activities, short-term memory. The motor cortex is responsible for voluntary movement and impulse control. Again, development of this frontal lobe helps us control our impulses when we're mature, hopefully. Okay, parietal lobe here on the top rear of the brain. It's a location for visual attention, location for touch perception. Goal-directed voluntary movements are found here. Manipulation of objects, physical manipulation of objects. Integration of several different senses that allows for understanding of a single concept. Moving on to the temporal lobe, this is right above your ear. Your ability to hear, memory acquisition, some visual perceptions, but this isn't the main function of it. Categorization of objects. Occipital lobe, vision. So this whole occipital lobe on the posterior part of the brain is just for interpreting vision because our eyes are so detailed, because visual stimulus is so needy of an entire region to process it. The occipital lobe is just for vision. The cerebellum, which is sometimes called the brain below the brain, what is it for? It's for coordination of voluntary movement, basically the storage of repetitive actions. So for example, if you've learned how to play the piano, you don't have to think very much about moving your fingers. That's because the information for how to move your fingers are stored in the cerebellum, and you don't have to think of it consciously if you've done it many times in the past. Helps with balance and equilibrium. Some memory for reflex motor acts like swallowing. The brainstem is found in the center here, and you can see it over here. Includes the midbrain, pons, and medulla. What are the functions of the brainstem? Well, breathing, centers for breathing control, heart rate, swallowing, reflexes for seeing and hearing, the startle response. Controls sweating, blood pressure, digestion, body temperature. So you see the brainstem is really the part of your brain that keeps you alive, the lizard brain, so to speak, that is responsible for all the basic decisions, let's say, of keeping you alive. Affects level of alertness, wakefulness, or drowsiness. Your ability to sleep is here to a certain extent. And your sense of balance is found in here. The vestibular function is coordinated there. Your midbrain, what is the function of it? It helps you with body posture, equilibrium, contains the autonomic nervous system, helps with blood pressure responses, temperature responses, like maintaining body temperature, and emotional influence, rage, and things like that. It can also regulate appetite and hormonal levels. And contains the nuclei of cranial nerves 3 and 4. These are Roman numerals, by the way, 3 and 4. The pons, its main function is for controlling respiration, chewing reflexes, taste, arousal, wakefulness, and alertness. Contains the nuclei of cranial nerves 5, 6, 7, and 8. The medulla, the lowest part and most primitive part of the brain, its functions are life-sustaining control centers, controlling hearing, respiration, and vasomotor, the vasodilation or vasoconstriction of blood vessels. Coughing, gagging, swallowing, vomiting, and digestive reflexes. Contains the cranial nerves of, contains the nuclei of cranial nerves 8, 9, 10, 11, and 12. That's how you count in Roman numerals. What are some functions of the different hemispheres? Well, the right hemisphere is primarily involved in judging the position of things in space, knowing body position, like where your arms and legs are, understanding and remembering things you do and see, putting bits of information together to make an entire cohesive picture, controls the left side of the body. So this is interesting. The right side of your brain controls the left side of your body. The left hemisphere, understanding the use of language, listening, reading, speaking, and writing, sometimes considered the more logical side of the brain. Memory for spoken and written messages, detailed analysis of information. So if somebody says they're a left brain individual, it means they're really good at analysis. Controls the right side of the body in the same way the other side did. So this shows you the different parts of the surface of the brain, especially the lateral surface. You can see the lateral fissure. It's this long line here. Each of these little folds and wrinkles are called gyrus. One is a hill. Sulcus is a little indentation. You see the different lobes here. This shows the inferior view, showing all the cranial nerves. Here's the pituitary gland, which is actually connected to the bottom of the brain through the infundibulum, pituitary stalk. Mammillary bodies are here, and you can see the olfactory bulbs here, which go directly down into your nose, into the olfactory nasal cavity, contains the olfactory epithelium. This is a sagittally section lateral view, showing you the cross section of the brain. Corpus callosum, which is a connective between left and right sides. Interthalamic adhesion connects left and right thalamus. There's a cerebrum showing you the mesencephalic aqueduct. There's the pons, medulla oblongata, containing the cerebral nuclei. There's basically an overview for you. Organization of brain tissue. Gray matter houses motor neuron and interneuron cell bodies, dendrites, axon terminals, and unmyelinated axons. It is gray because it does not have the myelin that the axons have that would make it white matter. White matter is composed primarily of myelinated axons. During brain development, an outer superficial region of gray matter forms from migrating peripheral neurons. External sheets of gray matter called the cortex cover the surface of most of the adult brain, including the cerebrum and cerebellum. So the outermost part is called the gray, the inner part is white. Organization of brain tissue. White matter lies deep to the gray matter of the cortex. Now this is in the brain. Spinal cord is going to be reversed. Within the masses of white matter, the brain also contains discrete innermost clusters of gray matter called cerebral nuclei, which are oval, spherical, or sometimes irregularly shaped clusters of neuron cell bodies. Remember, the cell bodies are not going to be surrounded by myelin and therefore will be gray in staining. So if you do a section or cut through these different parts of the brain, this is a frontal coronal section. We have two transverse sections through this cerebellum and through the medulla. So here's what we're looking at in the coronal section, showing you the white matter in the center, the lateral ventricles, which are filled with cerebrospinal fluid, and the gray matter on the outside. Now, if we move downwards, we can see what it looks like in the cerebellum. Again, gray matter on the outside, white matter on the inside. tube containing cerebrospinal fluid is smaller now, and right here in the center, moving downwards through the medulla oblongata, you see things starting to switch. And then all the way down in the spinal cord, we've reversed from gray matter on the outside and white inside to white on the outside, gray on the inside. So it switches right about in the area of the medulla oblongata. Support and protection of the brain. The brain is protected and isolated by multiple structures. The bony cranium provides rigid support, which we're talking about in your skull. Protective connective tissue membranes called the meninges surround and partition portions of the brain. Those meninges become inflamed, you have what is called meningitis. My brother almost died of meningitis when he was a baby. Cerebral spinal fluid, or CSF, acts as a cushioning fluid in which your brain floats, believe it or not. Your brain is actually floating inside your skull in a very thin bath of CSF. The brain has a blood-brain barrier that I mentioned earlier to prevent entry of harmful materials from the bloodstream into your brain. The meninges. There's 3 dense regular connective tissue layers that separate the soft tissue of the brain from the bones of the cranium. They enclose and protect blood vessels that supply the brain. They contain and circulate cerebrospinal fluid, or CSF. Parts of the cranial meninges form some of the veins that drain blood from the brain. These would be called sinuses. Sinuses, we saw them in the heart. They are just really large open spaces containing large amounts of blood. From superficial to deep, the cranial meninges are the dura mater on the outside, the arachnoid in the middle, and the pia mater directly attached to the surface of the brain. Here you can see the superior sagittal sinus. There's venous blood trapped here. And arachnoid villus are places where cerebrospinal fluid leaves this arachnoid layer, goes out and back and rejoins the blood. These are different layers of this region of your skull. The dura mater. It's a tough membrane composed of 2 fibers later. Think of durable dura mater. The strongest, toughest of the meninges. The dura mater is composed of 2 separate layers. The periosteal layer, the more superficial layer, attaches to the periosteum of the cranial bones. It really almost forms the periosteum of the cranial bones. The meningeal layer lies deep to the periosteal layer below it. The meningeal layer is usually fused to the periosteal layer, except in specific areas where the 2 layers separate to form large blood-filled spaces called the dural venous sinuses. And so it's basically a separation between those 2 layers where blood can move through your brain. The arachnoid. I don't know if you've ever heard of arachnophobia. It's a fear of spiders. It's called the arachnoid because when they first looked at it, it looked like spider webs. And that's the name, hence the name. Also called the arachnoid mater, or the arachnoid membrane, lies immediately internal to the dura mater. Partially composed of a delicate web of collagen and elastic fibers termed the arachnoid trabeculae. Now we're going to see trabeculae in the spongy bone found at the end of long bones, or the inside of your heart. Have these things called trabeculae, which are kind of fibrous extensions of the tissue. Between the arachnoid and the overlying dura mater is the subdural space. You may have heard of a subdural hematoma or a epidural. It has to do with the location of the dura mater. Immediately deep to the arachnoid is the subarachnoid space, and this is where you find those fibrous material that allows the blood vessels to penetrate across the surface of the brain. The pia mater is the innermost of the cranial meninges and forms a skin around the outside of your brain. It's a thin layer of delicate connective tissue that tightly adheres to the brain, follows every contour of the brain surface. So it's like a brain skin. Cranial dural septa, as I mentioned, the meningeal layer of the dura mater extends as flat partitions, or septa, separations, deep into the cranial cavity at 4 locations called the cranial dural septa. Now the septa separate, or as think of the term septa, separates the brain into different sections. The membranous partition separates specific parts of the brain. and provide additional stabilization and support to the entire brain. The falx cerebri, the tentorium cerebelli, the falx cerebelli, and the diaphragma celli. So this shows you, this is the big one. This is the divider called the falx cerebri, which divides the cerebral hemispheres in half. Down here, we can see the falx cerebelli that divides left and right halves of the cerebellum, excuse me. Then we have the tentorium cerebelli, which separates the bottom of the cerebrum from the cerebellum. And the diaphragma celli is a tiny little connective tissue layer that separates the pituitary from the inferior part of the midbrain. This is what it looks like in a cadaver. The ventricles of the brain. Now, in the same way that your heart contains blood, the ventricles of the brain contain a fluid called cerebrospinal fluid. Cavities are expansions within the brain that are derived from the lumen or opening of the embryonic neural tube. They're continuous with one another as well as with the central canal of the spinal cord, a little tube running down the spinal cord. There are 4 main ventricles in your brain. There are 2 lateral ventricles, which represent numbers 1 and 2. In the cerebrum, separated by a thin median partition called the septum pellucidum. Within the diencephalon is a smaller ventricle called the third ventricle. So that's third. Each lateral ventricle communicates with the third ventricle through an opening called the interventricular foramen. And last, the fourth ventricle is located within the pons and cerebellum. It's the last swelling open space as you move your way down into the spinal cord. And this shows you a model, if you could look through the brain. Lateral ventricles, 1 and 2. Third ventricle, right in the center here. If you remember from the previous image, the interthalamic adhesion would be right here in the middle. These lateral ventricles go all the way down the sides. Then we have what is called the mesencephalic aqueduct. Mesencephalic means in the middle of your head. Aqueduct means something conducts fluid. So the fluid is going to move downward from here to the fourth ventricle, which is just anterior to the cerebellum. And then the median aperture allows some fluid out. Then the fluid also contains down to the central canal of the spinal cord. There's the anterior view of the same thing, showing 3-dimensional shape of the lateral ventricles. Cerebrospinal fluid. It's a clear, colorless liquid that circulates in the ventricles and subarachnoid space. It bathes the exposed surfaces of the central nervous system and completely surrounds it. So as I told you, your brain is floating in a very small amount of cerebrospinal fluid. Performs several important functions. Buoyancy, lifting your brain up. Protection, so that your brain doesn't slam into the cranium. Environmental stability helps make sure that tissue changes, chemical tissue changes don't happen quickly in those tissues. Cerebrospinal fluid is formed by the choroid plexus in each ventricle, usually shown as a pink structure. This is a place where the blood vessels are leaky, allowing fluid, it's called cerebrospinal fluid, very similar to blood plasma, by the way. The noise you hear in the background is a puppy trying to dig into his food bowl, get more food out of it. So there you go. It's produced by a secretion of a fluid from the ependymal cells that originates from blood plasma, as I said. It's very similar to blood plasma, so I'm giving you a preview to the notes here. Now, how does the CSF make its way back into the blood? Well, in the subarachnoid space, it comes into these places called arachnoid villi. In this case, it's 1 or villus. You see this little tiny gap here, allows the fluid to move between these layers and back into the venous blood. This figure shows the movement of CSF from the choroid plexus, which is both here and here, by the way, and it moves its way down through the mesencephalic aqueduct into the fourth ventricle, through the median aperture, going around the outside of the brain, and then eventually all the way down through the central canal. But ultimately, it makes its way into the arachnoid villi and back into the venous blood. The blood-brain barrier, been mentioned a couple of times. Nervous tissue is protected from the general circulation by the blood-brain barrier. It strictly regulates what substances can enter the interstitial fluid of the brain. It prevents exposure of neurons in the brain to drugs, waste products of the blood, and variations in levels of normal substances, such as ions or hormones, that could adversely affect brain function. Tight junctions of the blood-brain barrier prevent materials from diffusing across the capillary wall. Astrocytes, which I showed you earlier, act as the gatekeepers that permit materials to pass to the neurons after leaving the capillaries. So these are a secondary structure that makes sure that only the substances that are supposed to get to brain cells do. It's markedly reduced or missing in 3 distinct locations in the central nervous system: choroid plexus, hypothalamus, and the pineal gland. The reason for this I'll be talking about in great detail in physiology. So this shows you how the astrocytes wrap around the capillaries, forming the blood-brain barrier. These show you the different lobes of the brain: frontal, parietal, occipital. This shows you an actual human brain with some of the same structures. These show you some functional regions of the brain that I'm not going to go into in a lot of detail, but if you're interested, take a look. Visual areas, premotor, frontal eye field, things like that. There's our homunculus again, showing you the different regions of the brain and how they're mapped across the surface of the cerebrum. This shows you that area of the central part of the brain, cross-section, showing you some of the key structures I pointed out before. Pineal gland, which produces melatonin for sleep-wake cycles. A whole bunch of other structures here you can get more information about in your text. This shows you the hypothalamus and some of these nuclei found in the hypothalamus, as well as the pituitary gland that's attached to the hypothalamus through the infundibulum. This is more of the inferior view of the brain that I showed you before, with the cranial nerves coming out the bottom. And even more detail here showing more cranial nerves. This is a cadaver's brain showing the same cranial nerves. Okay, that is 15. So, last but not least, I know it seems a lot, but we're going to go to 16 now. We'll be done for the day. Spinal cord and spinal nerves. Okay, here we go. Spinal cord provides a vital link between the brain and the rest of the body. Exhibits some functional independence from the brain. Some decisions that your body makes are made in the spinal cord and not the brain. Spinal cord and its attached spinal nerve serve 2 important functions. It's a pathway for sensory and motor impulses and contains areas that are responsible for reflexes. So when you take my physiology class, we'll be doing a reflex lab, and those reflex centers are found within the spinal cord. What is the structure of the spinal cord? Well, a typical adult spinal cord ranges between 42 and 45 centimeters in length. Now, if you do the math on that, you realize that your spinal cord proper ends right at the bottom of your rib cage. And that's not the full length of your back. So what's below your rib cage if it's not your spinal cord? Viewed in cross section, it's roughly cylindrical, but slightly flattened both posteriorly and anteriorly. Its external surface has 2 longitudinal depressions. The posterior or dorsal median sulcus dips internally in the posterior surface, and even deeper, the anterior or ventral median fissure is observed on its anterior surface. So the sulcus is smaller, fissure deeper. What is the different regions of the spinal cord? The cervical region is the superior-most region of the spinal cord. It's the thickest part of the spinal cord because fewer nerve fibers have branched out at that point. It's continuous with the medulla oblongata, contains neurons whose axons form the cervical spinal nerves. The thoracic region Do that again. The thoracic region lies inferior to the cervical region. Attached to this region are the thoracic spinal nerves. So this is your chest, where your ribs are. The lumbar region is a shorter segment of the spinal cord that contains neurons for the lumbar spinal nerves. Now, in the lumbar region, there is no spinal cord proper, and I'll show you what's there in the image. The sacral region lies inferior to the lumbar region and contains the neurons for sacral spinal nerves. The coccygeal region is the inferior tip of the spinal cord. One pair of coccygeal spinal nerves arises from this region. Spinal cord is shorter than the vertebral canal that houses it. I mentioned that earlier. Why is that the case, and what is the benefit? Well, I will tell you this. My son broke his back when he was 16, and if his spinal cord had been 2 or 3 millimeters longer, he would have been completely paralyzed. But because it ended at the bottom of his thoracic vertebrae, he had trauma, lost a vertebrae, but he's still able to walk. It was a miracle that it happened right there. And it had to do with the fact that there was no spinal cord proper in the place where he broke his back. The tapering inferior end of the spinal cord is called the conus medullaris, which means middle cone. It marks the official end of the spinal cord proper, that thick tubular structure that makes up the spinal cord. Inferior to this point, nerve roots, which are groups of axons collectively called the cauda equina. Now, in Latin, this means tail of the horse. Tail of the horse. Project inferiorly from the spinal cord. And that's what runs through the spinal canal or vertebral canal below the bottom of your thoracic vertebrae. Within the cauda equina is the filum terminale, a thin strand of pia mater that helps anchor the conus medullaris to the coccyx so that your spinal cord doesn't move up towards your brain. Spinal cord is associated with 31 pairs of spinal nerves that connect the central nervous system, the muscles, receptors, and glands. Each side of the spinal cord contains 8 cervical nerves called C1 to C8, 12 thoracic nerves, T1 to T12, 5 lumbar nerves, L1 to L5, 5 sacral nerves, S1 to S5, and 1 coccygeal nerve, which is C0. So this is what your spinal cord looks like. So we have cervical, purple, thoracic, orange. The note right here, the conus medullaris, is the end of the spinal cord proper. Everything below this is just spinal nerves, roots. Because my son broke his back right here, it didn't sever the spinal cord proper. Instead, the nerve roots moved out of the way so that he could be put back together and he's still walking. It's quite the miracle. Okay, here's the spinal cord, posterior median sulcus. You see the posterior side is more like just a slight indentation. And you see the posterior rootlets here. This is the end of the spinal cord. You can see the filium terminale, cauda equina, or horse's tail. This is the actual cadaver. And the dura mater has been opened up. Spinal meninges. They're continuous with the cranial meninges, same ones, same places. The structures that encircle the spinal cord, listed from outermost to innermost, are, well, the vertebrae is a protective structure. Then you have the epidural space where you would have an epidural shot to deaden you if you were going through childbirth. Then the dura mater, then the subdural space, then the arachnoid, then the subarachnoid space. Again, you find those kind of spiderwebby things. Then the pia mater would be attached directly to the spinal cord. This is a cross-sectional image showing you the same structure. Another image showing you the white matter, gray matter, the posterior and anterior roots, and how they articulate with the spinal cord, and a little bit of the connective tissue, dura mater here. Okay, location and distribution of the gray matter. In the spinal cord, gray matter is centrally located. This is the opposite of the brain, where it's outermost located. So superficially located versus deep. Its shape resembles the letter H or butterfly. Gray matter can be subdivided into the following components. So you can have anterior horns, lateral horns, and posterior horns. So front, side, back. There's also the gray commissure, which is a communicative structure that links the left and right side of the gray matter of the spinal cord. Location and distribution of the white matter. The white matter of the spinal cord is external to the gray matter. The white matter on each side of the cord is also partitioned into 3 regions. A posterior fenunculus lies between the posterior gray horns on the posterior side of the cord and the posterior median sulcus. Why is it called a fenunculus? Well, a fenunculus travels up and down a mountain, and so this region travels up and down your spinal cord, having that same term, fenunculus, connection between what's up high and down low. White matter region on each lateral side of the spinal cord are the lateral fenunculus. The anterior or fenunculus or fenunculi are composed of tracts of white matter that occupy the space on each anterior side of the cord between the anterior gray horns and the anterior median fissure. The anterior fenunculi are interconnected by the white commissure. White commissure, like the gray commissure, connects left and right sides of the spinal cord. This shows you some of the key structures. We see the gray matter looking like the letter H or a butterfly, and the white matter around it with some of the horns, fenunculi, and commissures shown here. Here's an actual stained slide of a human spinal cord. Gray matter in this kind of brighter purple, and the white matter in the darker. Spinal nerves. 31 pairs of spinal nerves connect the central nervous system to muscles, glands, and receptors. Each spinal nerve is formed from the union of thousands of motor and sensory neurons, axons. Motor axons originate from the spinal cord. Each anterior root and its corresponding posterior root unite within the intervertebral foramen to become a spinal nerve. The intervertebral foramen is formed from the upper vertebrae having a structure like this and the lower one having this. So when they come together, they form a whole or foramen. They contain both motor axons and sensory axons. What I want you guys to think about is that in the same way that the freeway has both northbound and southbound traffic, most nerves, almost all of them, as a matter of fact, contain both sensory and motor axons, so going up and down through the same nerve. Each spinal nerve is associated with the vertebra of the same number. Another image showing you some of the muscles of the back and the spinal cord with its associated ganglia and nerves. A dermatome. What is a dermatome? Well, it's a specific segment of skin supplied by a single spinal nerve. All spinal nerves, except for C1, innervate a segment of the skin, and so each of these nerves is associated with a dermatome, or region of the skin. The skin of the body may be divided into sensory segments that collectively make up a dermatome map. And this is the dermatome map. So interestingly, you can see C2 and C3 innervate right here around the chin, C4 here. Then look at C5 and C6 go all the way down your arm. So if you break your neck below the cervical vertebrae between the thoracic and cervical, you retain most of the use of your arm because the arms are innervated by cervical neurons or nerves, whereas the thoracic primarily innervate the chest. There's the lumbar going all the way down the legs. Sacral, coccygeal. Nerve plexuses. They're a network of interweaving anterior rami spinal nerves. The anterior rami of most spinal nerves form nerve plexuses on both the right and left sides of the body. The nerve plexuses then split up into multiple named nerves that innervate various body structures. So before the plexus, they're not named. For example, your radial nerve or ulnar nerve, those only get those names as they move away from the plexuses. The principal plexuses are the cervical, brachial, lumbar, and sacral. Now, which one of these is different than the others? Well, cervical, lumbar, and sacral all correspond directly to regions of the vertebrae. The brachial is talking about your arm, so this one's different. So instead of thoracic, we call it brachial. So this shows you some of the nerves and the plexuses associated with those nerves. More plexuses and nerves. Notice that they're not named nerves until they become yellow. Here, the joining and splitting of these rami become the plexuses. And this is what it looks like in a cadaver. This shows you some of the named nerves associated with the arm. These are more of the plexuses and nerves associated with the lumbar region and what you would see in a cadaver. These are the named nerves in the leg. These are the plexuses and nerves associated with the sacrum. The sciatic nerve, being one of the bigger ones, is actually associated with this tibial division. So we have two divisions of the sciatic nerve, fibular and tibial division. They're showing this as blue, but really it is a named nerve at this point, so it should be the sciatic nerve and should be all yellow. This shows you some of the posterior nerves of the leg and what those nerves would look like in a cadaver. Reflexes. What is a reflex? It's a rapid, automatic, involuntary reaction of muscles or glands to a stimulus. So in the same way that if a doctor taps you under the kneecap, pokes you towards the eye, you'll blink, it's a reflex, something you do automatically. All reflexes have similar properties. First, you have to begin with a stimulus required to initiate a response to sensory input. Something has to happen. A rapid response requires that few neurons be involved and a synaptic delay be minimal. The key is you want it to go in and out quickly so you can respond quickly to this stimulus. An automatic response occurs the same way every time, so it's not something you have to think about. An involuntary response requires no intent or pre-awareness of the reflex activity. So often your reflex can be initiating at the same time you even know it's happening. Your body has made that decision to start the reflex even before you know it has started. Reflexes are usually not suppressed. Now, some suppressed reflexes are things like the reflex to urinate, to micturate. We repress that as post-toilet trained kids because we don't want to soil our pants. In animals like horses, they don't suppress it at all. When they got to go pee, they just go wherever they are. But animals that have a home or a den tend to suppress it so that they don't soil that den. And they can also use that urine to mark their territory. That's one of the reasons why urinary reflexes are suppressed in hunting animals primarily. Awareness of the stimulus occurs after the reflex action has been completed in time to correct or avoid a potentially dangerous situation. So reflex happens, you find out about it, then you can act in a conscious way to avoid any further damage. What are the components of a reflex arc? It's the neural wiring of a single reflex. It always begins at a receptor in the peripheral nervous system. There's communication with the central nervous system, and it ends at a peripheral effector, usually a muscle, sometimes a gland. There's two types of reflex arcs that we're going to talk about, ipsilateral and contralateral. Ipsilateral is when both the receptor and effector organs of the reflex are on the same side of the spinal cord. For example, an ipsilateral effect occurs when the muscles in your left arm contract to pull your left hand away from the object. Also, that patellar stretch reflex, when someone hits you under the kneecap, your leg goes out. Stimulus and effect are on the same side. Contralateral is when the sensory impulses from a receptor organ cross over through the spinal cord to activate an effector organ in the opposite limb. And this is where we get the crossover between white matter and gray matter. For example, contralateral effect occurs when you step on a sharp object with your left foot and then contract the muscles of your right leg to maintain balance. As you lift your left leg, to avoid stepping on the sharp object. A monosynaptic reflex is the simplest of all reflexes. Interneurons are not involved, so it goes directly from sensory to motor. The patellar or knee-jerk reflex is a monosynaptic reflex that physicians use to assess the functioning of the spinal cord. By tapping the patellar ligament with a reflex hammer, the muscle spindles in the quadricep muscles are stretched. That triggers a response to straighten the leg. produces a noticeable kick of the leg, which is your body's attempt to straighten your leg to keep you from falling down. It doesn't know you're sitting down. It's a reflex. Polysynaptic reflexes have more complex neural pathways that exhibit a number of synapses involving interneurons within the reflex arc. Because this reflex arc has more components, there's a more prolonged delay between stimulus and response. So as you can see here, this is a monosynaptic reflex. The sensory receptor is stimulated. It goes directly from sensory to motor, quick response in the kicking of the leg. But in a polysynaptic reflex, you have the sensory, have the interneuron, and the motor neuron. The interneuron has to make some decisions. What are we going to do with this information? We're going to send it out to just one set of muscles or multiple? We have to recruit other muscles that aren't directly associated with it so we don't fall down. That's what the interneuron is for. It's a neuron that makes a decision. Okay, stretch reflexes like the patellar stretch reflex. It's a monosynaptic reflex that monitors and regulates skeletal muscle length. When a stimulus results in the stretching of a muscle, that muscle reflexively contracts to keep the muscle at normal length. The patellar or knee jerk reflex is an example of a stretch reflex. The stimulus, which is the tap on the patellar tendon, initiates contraction of the quadriceps femoris muscle and extension of the knee joint. Now, this would be useful if you're standing up and your knee buckles. Well, your quadriceps can stretch quickly. You didn't intend on that happening. So the reflex is to contract the quadriceps muscle. You stand back up. You may have noticed that on occasion. You're standing there, your knee buckles, and you just pop up really quick. That's part of that patellar stretch reflex, keeping you from landing on your butt. This shows you the structures of the muscle spindle, so you can sense whether or not the muscle's been stretched. Golgi tendon reflex prevents skeletal muscles from tensing excessively. Golgi tendon organs are nerve endings located within tendons near a muscle tendon junction. Activation of the Golgi tendon organs signal interneurons in the spinal cord, which in turn inhibit the actions of the motor neurons. So if you're contracting muscle too hard, you're going to damage it. So this signal says, back off, you're going to hurt yourself if you pull too hard. The associated muscle is allowed to relax, thus protecting the muscle and tendon from excessive tension damage. And there's the sensor that we sense whether there's too much tension on the muscle. Reflex testing in a clinical setting. Reflexes can be used to test specific muscle groups and specific spinal nerves or segments of the spinal cord. Consistently abnormal reflex response may indicate damage to the nervous system or the muscles, anything in the pathway. The reflex response may be normal, hypoactive, which is underactive, hyperactive, which is overactive. Okay, that is the end of chapter 16. Kiara, do you have any questions or do you have any review questions that you'd like to go over for the exam? Okay, not getting anything from Kiara, so I'll take that as a no. That's it for today. Good luck with your exam this week, and hope to see you back next time for more chapters. Have a good rest of your week, folks. Bye now.